Welcome back to Manifest Masters. Tonight, we're not going anywhere. We don't need to. Because the most extraordinary place you will ever encounter is not a city buried under 2,000 years of history. It is not a pyramid rising from the desert floor or a temple carved into a cliff face or a library full of the accumulated knowledge of the ancient world. It is the thing you are already inside. The thing that has been carrying you through every moment of your life without asking for your attention or your gratitude or even your awareness of what it is doing on your behalf. Your body. Right now, while you listen to these words, while your eyes grow heavy and your breathing slows and the day begins its long retreat, your body is doing things so complex, so precise, so quietly astonishing that if you could see them happening you would not believe they were possible. And they are happening in you. In the specific, particular, irreplaceable you that has never existed before and will never exist again. So settle in. Let your body go heavy. And let it tell you its story. Begin with the breath. Not because the breath is the simplest place to start, it is not, as it turns out, simple at all, but because it is the most immediate. The most present. The thing that is happening right now, as these words arrive, the thing that has been happening since the first moments after you were born and that will continue, without any instruction from you, for as long as you are alive. You are breathing right now. You were breathing before you started listening and you will be breathing after you have fallen asleep and you will be breathing in the deep middle of the night when consciousness has retreated entirely and the world has narrowed to the slow rise and fall of your chest in the dark. You take somewhere between 17,000 and 23,000 breaths every single day. Not consciously, you are not deciding to take each one, not counting them, not managing them. Your brainstem is doing that. The most ancient part of your brain, the part that you share with creatures whose ancestors were navigating the world hundreds of millions of years before the first human being drew breath, is monitoring the carbon dioxide levels in your blood with continuous, tireless precision and adjusting the rate and depth of your breathing accordingly, moment by moment, so automatically and so reliably that you can go your entire life without ever once thinking about it and it will never miss a single breath on your behalf. The air that enters your lungs with each of those 17,000 daily breaths travels down through the trachea and into the bronchial tubes and from there into the lungs themselves, which are not the simple balloon-like structures that a diagram might suggest but something far more intricate, a branching network of progressively smaller tubes, dividing and subdividing like the branches of an enormous tree, ending eventually in tiny air sacs called alveoli whose walls are so thin that oxygen can pass directly through them into the bloodstream. If you could unfold all of the alveoli in both of your lungs and spread them out flat, the total surface area they represent would cover somewhere between 70 and 100 square meters. The internal surface of your lungs is roughly the size of a singles tennis court, folded with extraordinary precision into a space not much larger than your two fists held together. In that space, in those 100 square meters of folded surface, your blood picks up oxygen and releases carbon dioxide approximately once every second. 24 hours a day, 7 days a week. For the entire length of your life, without pause, without rest, without any conscious participation from the person whose life it is sustaining. Your heart, meanwhile, is doing something equally extraordinary in the space just beside your lungs. It is beating. Right now, as you read these words, your heart is contracting and relaxing in a rhythm so consistent and so reliable that its disruption is one of the clearest signals the body can send that something has gone wrong. The average human heart beats around 70 times per minute at rest. That is 4,200 times per hour. 100,000 times per day. 37 million times per year. And over the course of an average human lifetime, approximately 70 to 80 years, your heart will beat somewhere in the region of 2.5 to 3 billion times. 3 billion contractions, each one a precisely coordinated squeeze of 4 chambers working in sequence, each one moving blood through a network of vessels so extensive that if you could remove every blood vessel from an average adult human body, every artery and vein and capillary, and lay them end to end in a single line, that line would stretch for approximately 100,000 kilometers. 2.5 times around the circumference of the Earth. All of it inside you. All of it right now. The blood that moves through those 100,000 kilometers of vessels is itself something worth pausing over. It is not simply a red liquid, it is a living tissue, one of the few liquid tissues in the human body, containing several distinct types of cells suspended in a pale yellowish fluid called plasma. The red blood cells, the ones that give blood its color, the ones that carry oxygen from the lungs to every corner of the body and carbon dioxide back again, are among the most specialized cells in the human body. They are so specialized, in fact, that they have no nucleus. In order to maximize the space available for hemoglobin, the protein that binds oxygen and gives red blood cells their oxygen-carrying capacity, the cells eject their own nuclei during development, sacrificing their ability to reproduce and repair themselves in exchange for the ability to carry more of what the body needs. A cell that gives up its own genetic material in order to do its job better. A biological act of total commitment, repeated in the bone marrow somewhere between 2 and 3 million times every single second of your life, as old red blood cells are destroyed and new ones are produced to replace them. 2 to 3 million new red blood cells every second. Right now, while you rest. The white blood cells that travel alongside the red ones in the bloodstream are doing something entirely different. Where red blood cells are simple, highly specialized, devoted entirely to a single function, white blood cells are complex, various, and engaged in the continuous and enormously demanding work of keeping you alive in a different sense, not by delivering oxygen but by defending the body against the bacteria and viruses and fungi and parasites and malfunctioning cells that are constantly, in every moment of your life, attempting to establish themselves in the warm and nutrient-rich environment your body provides. Your immune system, the network of cells and proteins and organs that coordinates this defense, is capable of recognizing and responding to an almost unimaginable variety of threats. It is estimated that the human immune system can produce antibodies, the specialized proteins that bind to and neutralize specific pathogens, for somewhere in the region of 10 billion distinct molecular targets. 10 billion. The variety of threats the immune system is prepared to encounter and defeat is so vast that it exceeds the number of distinct pathogens that have ever existed in the history of life on Earth, which means your immune system is prepared for threats that have not yet evolved. It is defending you against the future. And it is doing all of this right now. While you rest. While you breathe. While your heart beats its 3 billionth beat and your lungs fold and unfold their tennis court of alveoli and your bone marrow manufactures its 2 million red blood cells per second and you lie here, entirely unaware of any of it, letting the night hold you in its quiet dark. The brain that is allowing you to do all of this, that is processing these words and generating the images they produce and managing your breathing and your heart rate and the temperature of your body and the position of your limbs and 1,000 other variables simultaneously, is the most complex structure that has ever been discovered anywhere in the known universe. It contains approximately 86 billion neurons, individual nerve cells, each one connected to thousands of others through junctions called synapses, creating a network of connections so vast that the total number of synapses in a single human brain is estimated at 100 trillion. 100 trillion connection points, each one capable of transmitting signals at speeds of up to 430 km per hour, the whole network operating simultaneously, continuously, generating in its collective activity the thing we call thought. The thing we call consciousness. The thing we call you. The brain that contains all of that weighs approximately 1.4 kilograms. It has the consistency of soft tofu. It uses about 20 watts of power, roughly the same as a very dim light bulb, to run the most sophisticated information processing system in the known universe. And it does all of this while consuming, despite being only about 2% of your total body weight, approximately 20% of all the energy your body produces. The brain is extraordinarily expensive to run, by the body's standards. And the body pays the price without complaint, every moment of every day, because the alternative is unthinkable. Your brain is running right now. It has been running continuously since before you were born. Fetal brain activity begins in the womb, the developing nervous system already processing and responding and learning long before the person it belongs to has taken a first breath. It will continue running until the last moment of your life. It has never been turned off. It has never had a moment of complete rest. Even in the deepest sleep, even in the dreamless hours when consciousness seems entirely absent, your brain remains active, processing the day's experiences, consolidating memories, clearing metabolic waste through a system of channels that becomes active specifically during sleep. The brain literally cleaning itself in the night while you rest, preparing itself for the demands of another day. Your body knows what it needs. It always has. And right now, in this moment, what it needs most is exactly what you are giving it. Rest. Let it have it. There is a conversation happening inside you right now that you have never heard and will never hear directly, conducted in a language so complex and so fast that the most powerful computers ever built can only approximate its simplest exchanges. It is the conversation your cells are having with each other. The continuous, simultaneous, extraordinarily precise signaling that coordinates the activity of the 37 trillion cells that make up a human body, keeping them informed of what is happening elsewhere, telling them what to produce and what to stop producing, when to divide and when to die, when to sound the alarm and when to stand down. 37 trillion cells. The number is so large that it resists genuine comprehension. If you counted 1 cell per second without stopping, it would take you more than 1,000,000 years to count them all. Each one is a living entity in its own right, taking in nutrients, producing energy, carrying out specialized functions, responding to signals from its environment, maintaining its internal chemistry within the narrow ranges that allow it to survive. Each one is more complex than any machine human beings have ever built. And all 37 trillion of them are working together, right now, in the body you are resting in, coordinated into a single coherent organism so seamlessly that you experience yourself not as a collection of trillions of individual living things but as one thing. One person. One continuous, unified experience of being alive. The coordination that makes this possible happens through several overlapping systems, each one communicating in its own way and at its own speed. The nervous system, the brain and the spinal cord and the vast network of nerves that branch from them into every corner of the body, communicates through electrical signals, fast and precise, capable of traveling from the brain to the tip of the toe in a fraction of a second. The endocrine system communicates through hormones, chemical messengers released into the bloodstream and carried to target cells throughout the body, slower than nerve signals but capable of producing effects that last hours or days rather than milliseconds. And at the cellular level, individual cells communicate directly with their neighbors through chemical signals passed across the tiny gaps between them, a molecular conversation happening simultaneously at 37 trillion points throughout the body, every moment of every day. The speed at which the nervous system operates is one of the things that most consistently astonishes people when they encounter the actual numbers. The fastest nerve fibers in the human body, the large myelinated fibers that carry signals related to touch and proprioception, the sense of where your body is in space, conduct signals at speeds of up to 70 to 120 meters per second. That is between 250 and 430 kilometers per hour. The signal that tells your hand to pull back from a hot surface travels from the pain receptors in your skin to your spinal cord and back to the muscles in your arm faster than conscious thought can register what is happening. You have already moved before you know you are moving. Your nervous system has assessed the threat, decided on a response, and executed it before the information has even fully reached the part of your brain responsible for conscious awareness. The body protecting itself before the mind has a chance to weigh in. This is possible because of a particular feature of spinal cord anatomy, the reflex arc, rather than routing the signal all the way up to the brain and back down again, which would take precious additional milliseconds. The spinal cord processes certain types of urgent signals locally, generating an immediate motor response without waiting for conscious authorization. The brain learns what happened afterward, receiving the information about the hot surface and the withdrawal of the hand as a report of something already completed rather than a request for instruction. The body has its own local intelligence, distributed through the spinal cord and the peripheral nervous system, capable of making certain decisions autonomously, without reference to the central authority of the brain. That distributed intelligence extends further than the spinal cord. The enteric nervous system, the network of neurons embedded in the walls of the gastrointestinal tract, running from the esophagus all the way to the rectum, contains somewhere between 200 and 600 million neurons. More neurons than are found in the spinal cord. More neurons than are found in the entire nervous system of a cat. This network operates largely independently of the brain, managing the extraordinarily complex process of digestion, the muscular contractions that move food through the gut, the release of digestive enzymes, the regulation of blood flow to the intestinal walls, the coordination of the various specialized regions of the gut that perform different digestive functions, with a degree of autonomy that has led some researchers to refer to the enteric nervous system as the second brain. Not metaphorically. Structurally. A second independent neural network, embedded in your gut, running digestion the way your brain runs thought, continuously, automatically, and largely without reference to anything else. The gut and the brain do communicate, however, through a channel that has become one of the most intensely studied areas of neuroscience in recent decades. The vagus nerve, the longest cranial nerve in the body, running from the brainstem down through the neck and chest and into the abdomen, carries signals in both directions between the brain and the gut, and the traffic on this nerve is considerably more one-sided than most people expect. Approximately 80% of the signals traveling along the vagus nerve are traveling upward, from the gut to the brain rather than from the brain to the gut. Your gut is sending far more information to your brain than your brain is sending to your gut. The feelings we describe as gut feelings, the physical sensations of anxiety or excitement or unease that manifest in the stomach and the intestines, are not merely metaphorical. They are the product of a genuine two-way conversation between two neural networks, in which the gut is, in terms of sheer signal volume, doing most of the talking. The microbiome that inhabits the gut and participates in this conversation is itself an entity of almost incomprehensible complexity. The human gut contains somewhere between 38 trillion and 100 trillion individual microorganisms, bacteria, fungi, viruses, archaea of several thousand distinct species, collectively carrying a quantity of genetic material that dwarfs the human genome. For most of the 20th century, these microorganisms were understood primarily as passengers, tolerated residents of the gut, neither particularly helpful nor particularly harmful in most circumstances. The research of the past 2 decades has fundamentally revised that understanding. The gut microbiome is now understood to be an active participant in human physiology, influencing immune function, metabolic processes, inflammatory responses, the production of certain vitamins and neurotransmitters, and increasingly, through the gut-brain axis, aspects of mood and behavior and cognitive function that were previously assumed to be entirely the province of the brain. The bacteria in your gut are producing neurotransmitters right now. Serotonin, the neurotransmitter most commonly associated with mood regulation, the one that most antidepressant medications work by modulating, is produced in the gut in quantities that dwarf the brain's own production. Approximately 90% of the body's total serotonin is produced not in the brain but in the gastrointestinal tract, by the enterochromaffin cells of the gut lining in response to signals from the gut microbiome. The chemistry of your mood is being conducted, in large part, in your intestines, by microorganisms that have been living in your gut since the first days of your life, that you have been feeding with every meal you have ever eaten, that have been shaping your biology in ways that science is only beginning to fully understand. Your skin, meanwhile, is doing something that tends to surprise people when they first encounter it. It is doing rather a lot. Skin is the largest organ in the human body, covering approximately 2 square meters of surface area and making up roughly 15% of total body weight in an average adult. It is not simply a passive wrapper, a biological container for the more impressive structures within. It is an active, metabolically busy organ performing several distinct and essential functions simultaneously. It is regulating your body temperature right now, through the dilation and constriction of blood vessels in the dermis and through the activity of sweat glands that can produce up to 10 liters of sweat per day under conditions of extreme heat or exertion. It is synthesizing vitamin D from ultraviolet radiation whenever it is exposed to sunlight, a process so essential to bone health and immune function and dozens of other physiological processes that vitamin D deficiency has become one of the most common nutritional deficiencies in the modern world, largely because modern lives are spent predominantly indoors. It is housing an immune system of its own, specialized cells in the epidermis called Langerhans cells that are constantly sampling the skin's surface for pathogens, presenting what they find to the immune system, and initiating responses to threats before they have a chance to penetrate deeper. And it is renewing itself. Constantly, continuously, without pause. The outermost layer of the skin, the stratum corneum, consists entirely of dead cells, cells that have completed their upward journey from the deeper layers of the epidermis and have lost their nuclei and their internal machinery and have become, in their final form, flattened envelopes of keratin whose sole remaining function is to form a physical barrier against the outside world. These cells are shed continuously. Somewhere between 30,000 and 40,000 dead skin cells fall from your body every hour. About 600,000 per day. In a year, you shed roughly the equivalent of your entire outer layer of skin, replaced cell by cell from below, the whole surface renewed without you ever noticing that it is happening. The dust in your home is, in significant part, made of you. The surfaces around you carry traces of a continuous quiet shedding that has been happening since the day you were born. You are leaving yourself behind, in the smallest possible increments, everywhere you go. And underneath that shed surface, underneath the extraordinary activity of the gut and the tireless signaling of the nervous system and the 100,000 kilometers of blood vessels and the tennis court of lung and the 3 billion heartbeats underneath all of it, the skeleton is doing something that almost nobody thinks about when they think about bones. It is making blood. The marrow inside your bones, the soft fatty tissue in the hollow interior of your long bones and the spongy interior of your flat bones is one of the most productive tissues in the human body. It is where red blood cells are born, where white blood cells are born, where platelets are born. The bone marrow of an average adult produces approximately 500 billion blood cells every single day. 500 billion. Not a year. A day. The skeleton that most people think of as a fixed and passive structure, the rigid framework that holds everything else up, is in fact a living, dynamic, continuously productive organ, generating the cellular components of blood at a rate that makes almost every other productive process in the body look modest by comparison. The bones themselves are not fixed and static either. They are in a constant state of renewal, old bone being broken down by cells called osteoclasts while new bone is built up by cells called osteoblasts, the two processes happening simultaneously throughout the skeleton, the balance between them determining whether bone density is increasing or decreasing at any given time in a person's life. The entire human skeleton is replaced, cell by cell, approximately every 10 years. The bones you have now are not the bones you were born with. They are not even the bones you had a decade ago. They are new, built from the materials your body has been given, shaped by the stresses and strains of the life you have been living, carrying in their density and their architecture a physical record of everything your body has been through. You are newer than you think, and more extraordinary than you have probably been told. Rest in that for a moment. Let the body do its work. It knows exactly what it is doing. Sleep is doing something to you right now. Or it is about to. If you are not already there, if you are still in that soft borderland between waking and sleep where the words arrive clearly but the images they produce are beginning to take on a quality that is slightly looser, slightly less tethered to the ordinary logic of the waking mind, then you are approaching it. Your body is preparing for it with the same systematic thoroughness it brings to everything, the same quiet efficiency that has been running your heart and your lungs and your immune system and your bone marrow without your knowledge or your help for every moment of your life. Sleep is not what most people think it is. The idea that sleep is simply the absence of wakefulness, a kind of biological standby mode, the body powering down its non-essential systems to conserve energy and recover from the demands of the day, is not wrong exactly, but it is so incomplete as to be misleading. Sleep is not a passive state. It is an active one. The sleeping brain is not a quieter version of the waking brain, it is a differently busy brain, engaged in processes that cannot happen during wakefulness, performing functions so essential that the consequences of their disruption become apparent with remarkable speed. A person deprived of food can survive for weeks. A person deprived of water can survive for days. A person deprived of sleep begins to show significant cognitive and physiological impairment within 24 hours, and the record for the longest a human being has voluntarily stayed awake 11 days, set by a 17-year-old American student named Randy Gardner in 1964, under medical supervision, ended with hallucinations, paranoia, and cognitive impairment severe enough that Gardner could not remember how to complete a simple counting task. Sleep is not optional. It is as fundamental to survival as food and water, and the body pursues it with the same urgency it brings to any other essential need, building pressure for it gradually across the waking hours through the accumulation of a chemical called adenosine in the brain, the increasing concentration of which produces the feeling we call tiredness, the signal the body sends when the need for sleep has become sufficiently urgent that it can no longer be comfortably ignored. The adenosine that accumulates during waking hours and drives the pressure to sleep is cleared during sleep itself, which is why a full night of sleep leaves you feeling refreshed. The chemical signal of tiredness has been literally washed out of your brain during the hours of sleep, the slate wiped clean, the pressure reset to zero. Caffeine, incidentally, does not give you energy. It works by blocking the receptors that detect adenosine, preventing the brain from registering how much of it has accumulated. The adenosine keeps building regardless, caffeine simply prevents you from feeling it building. When the caffeine wears off and the receptors become available again, all of the accumulated adenosine signals at once, which is why the crash that follows a period of caffeine use can feel so sudden and so comprehensive. Sleep itself is not a single uniform state but a structured cycle of distinct phases, each one characterized by different patterns of brain activity and different physiological processes. The cycle takes approximately 90 minutes to complete and repeats 4 to 6 times across a typical night, the composition of each cycle shifting as the night progresses, the early cycles dominated by deep slow-wave sleep, the later cycles dominated by REM sleep, the phase during which most dreaming occurs. Deep slow-wave sleep, the phase during which the brain produces the large, synchronized electrical waves that give it its name, is when the most physically restorative processes occur. Growth hormone is released in its largest daily pulse during deep slow-wave sleep, driving the repair and growth of tissues throughout the body. The glymphatic system, the brain's waste clearance mechanism, the network of channels that flushes metabolic byproducts from between brain cells, is most active during slow-wave sleep, the channels expanding to allow cerebrospinal fluid to flow through them more freely, washing away the proteins and metabolic waste products that accumulate during waking hours. Among the things cleared by the glymphatic system during sleep are the proteins associated with neurodegenerative diseases, amyloid beta and tau, the proteins whose accumulation in the brain is associated with Alzheimer's disease. Sleep is not merely restorative. In a very literal sense, it is protective, the brain cleaning itself of the molecular debris that, if allowed to accumulate, may contribute to the diseases of aging. REM sleep, rapid eye movement sleep, named for the characteristic darting movements of the eyes beneath closed lids that accompany this phase, is when the brain is at its most active during sleep, producing patterns of electrical activity that in some respects resemble waking more than they resemble the deep slow-wave phases that precede them. It is during REM sleep that most dreaming occurs, the brain generating vivid experiences that the dreamer typically accepts as real regardless of how implausible their content, the complete absence of critical evaluation that characterizes the dreaming mind, allowing scenarios of considerable absurdity to proceed without question. And it is during REM sleep that the brain performs one of its most important cognitive functions, the consolidation and integration of memories, the process by which the experiences of the day are sorted and evaluated and woven into the existing fabric of long-term memory, some experiences strengthened and some allowed to fade, the emotional content of memories processed and modulated, connections between apparently unrelated pieces of information sometimes formed in ways that can produce, upon waking, the experience of sudden insight, the solution to a problem that was unsolvable the night before arriving, seemingly from nowhere, in the first moments of waking consciousness. The dreams that accompany REM sleep are among the most mysterious and most studied phenomena in all of neuroscience, and despite centuries of human fascination with them and decades of scientific investigation, their function and their meaning remain genuinely contested. The activation synthesis hypothesis, proposed by the neuroscientists Hobson and Macaulay in 1977, suggests that dreams are essentially the brain's attempt to make narrative sense of the random patterns of neural activation that occur during REM sleep, the storytelling centers of the brain imposing structure on noise, generating the experience of a coherent narrative from inputs that are not themselves coherent. Other theories emphasize the role of dreaming in emotional processing, in threat simulation, in memory consolidation. What is not disputed is that REM sleep is essential. People selectively deprived of REM sleep while allowed to sleep normally in other respects show significant impairment in emotional regulation and memory consolidation, and the brain compensates for REM deprivation by producing more of it in subsequent nights, a phenomenon called REM rebound, as if the brain is aware of the debt that has been incurred and is determined to repay it. The body temperature that drops during sleep, falling by approximately 1 to 2 degrees Celsius from its daytime level in the hours after sleep onset, is not a side effect of inactivity. It is an active process, the body deliberately directing blood flow toward the skin to radiate heat away, lowering the core temperature to a level that facilitates the transition into and maintenance of deep sleep. This is why a warm bath before bed can help you fall asleep, not because it raises your body temperature, but because the rapid cooling that follows getting out of the bath mimics and accelerates the natural temperature drop that the body produces as it prepares for sleep. And it is why sleeping in a cool room tends to produce better sleep than sleeping in a warm one. The body's ability to maintain the lower temperature that facilitates deep sleep is easier when the environment is not working against it. The pineal gland, a small pinecone-shaped structure buried deep in the center of the brain, about the size of a grain of rice, is the body's primary timekeeper, the organ through which the body's internal clock communicates with the rest of the physiology to coordinate the timing of sleep and wakefulness. It does this through the production of melatonin, a hormone released in response to darkness and suppressed by light, whose rising concentration in the blood in the evening signals to the body that night is approaching and that the processes associated with sleep should begin. The pineal gland was described by the philosopher René Descartes as the seat of the soul, the point at which the immaterial mind interfaced with the material body, a designation that reflected both the gland's mysterious central location and the almost complete absence of any understanding of what it actually did. It is not the seat of the soul. But it is, in its own quiet way, one of the most important small structures in the body, a grain of rice-sized timekeeper whose output coordinates the sleeping and waking of every cell in the body with the cycle of light and dark in the world outside. Light suppresses melatonin with a specificity that has become increasingly relevant in an age of artificial illumination. The wavelengths of light most effective at suppressing melatonin production are in the blue end of the visible spectrum, the same wavelengths that are most abundantly emitted by the LED screens of phones and tablets and computers. The body's ancient light detection system, calibrated over millions of years of evolution to use the blue sky of daytime as a signal that it is not yet time to sleep, cannot distinguish between the blue of the afternoon sky and the blue light of a phone screen held 6 inches from the face at 11 o'clock at night. The screen tells the pineal gland that it is still day. The pineal gland delays the release of melatonin. Sleep is postponed, and the consequences accumulate across the days and weeks and years of a life lived in chronic mild sleep deprivation, in ways that research is only beginning to fully characterize. The body wants to sleep. It has always wanted to sleep. It has been wanting to sleep, in the most fundamental biological sense, for as long as there have been creatures complex enough to need it, which is longer than you might expect. Sleep, or something that functions like sleep, has been observed in virtually every animal species that has been studied for it, including animals with nervous systems far simpler than our own. Fruit flies sleep. Roundworms with exactly 302 neurons sleep. Even jellyfish animals with no brain at all, no centralized nervous system, just a diffuse network of nerve cells distributed through their bodies, show periods of reduced activity and reduced responsiveness that meet the behavioral criteria for sleep. Sleep did not evolve once in one lineage and spread from there. It evolved independently, multiple times, in organisms so different from each other that the only reasonable conclusion is that whatever sleep does, it does something so essential that evolution keeps arriving at it regardless of the starting point. Whatever sleep does, the body considers it non-negotiable. And right now, in this moment, your body is doing everything it can to help you get there. Let it. Stop resisting. The body knows the way. There is a place in the brain about the size of a grain of rice. We have already visited one of those tonight. The pineal gland with its melatonin and its ancient light detection system, but this one is different. This one is not a timekeeper. It is something closer to an alarm system. A threat detector. The structure that has been scanning your environment for danger since before you were old enough to understand what danger was, that continues scanning it now while you rest, that will be scanning it in the deepest hours of the night when consciousness has retreated entirely and you are as vulnerable as a human being ever gets. It is called the amygdala. There are two of them, one on each side of the brain, buried deep in the temporal lobes. Almond-shaped, the word amygdala comes from the Greek for almond, and they are among the most ancient structures in the human brain, present in something like their current form in the brains of animals whose lineage diverged from ours hundreds of millions of years ago. They are not sophisticated structures by the brain's standards. They do not engage in the nuanced analysis of complex situations that the prefrontal cortex specializes in. What they do is fast and simple and extraordinarily effective. They detect threat, and they initiate response, and they do both of these things faster than conscious thought can intervene. The fear response that the amygdala initiates, what most people know as the fight-or-flight response, more precisely called the acute stress response, is one of the most thoroughgoing physiological transformations the body is capable of producing. In the fraction of a second between the detection of a threat and the first conscious awareness of having detected it, the amygdala has already sent signals to the hypothalamus, the brain region that controls the autonomic nervous system, which has already begun orchestrating a body-wide response of remarkable coordination. The adrenal glands, sitting atop the kidneys, release adrenaline into the bloodstream. The heart rate increases. Blood pressure rises. The pupils dilate to admit more light. The airways in the lungs widen to allow more air. Blood is diverted away from the digestive system and toward the large muscles of the arms and legs. The liver releases stored glucose to provide immediately available fuel. Pain sensitivity decreases. Attention narrows to the source of the threat. All of this happens in seconds. The body goes from rest to full emergency readiness in the time it takes to draw a single breath, preparing itself with extraordinary efficiency for the two responses that evolution has found most useful in the face of immediate physical danger. Fight or flee. Hit or run. The options are ancient and the preparation for them is ancient, and the body executes it with the same precision it has been executing it for millions of years, regardless of whether the threat is a predator in the grass or a difficult conversation or a deadline or a noise in the house at night. The brain does not always distinguish very well between physical threats and psychological ones. The amygdala that evolved to detect a predator in the grass responds to social rejection and financial anxiety and anticipated embarrassment with much of the same neurochemical machinery it uses for genuine physical danger. The stress response that was designed to be brief and intense, deployed for the seconds or minutes of a genuine physical emergency and then rapidly resolved when the danger passed, can be triggered repeatedly and sustained chronically by the psychological pressures of a modern life in ways that the body was not designed to accommodate. Chronic stress, the persistent, low-level activation of the stress response by ongoing psychological pressures rather than immediate physical threats, has measurable effects on almost every system in the body. It suppresses immune function. It disrupts sleep. It damages the walls of blood vessels. It impairs memory and cognitive function. It accelerates cellular aging at the molecular level, shortening the protective caps on chromosomes called telomeres in a way that has been directly correlated with the experience of chronic psychological stress. The telomere connection to stress is one of the more striking findings in recent biology. Telomeres are the repetitive sequences of DNA that cap the ends of chromosomes, protecting them from deterioration the way the plastic tips on shoelaces protect the laces from fraying. Every time a cell divides, the telomeres shorten slightly. This shortening is understood to be one of the fundamental mechanisms of cellular aging, cells becoming less capable of accurate replication as their telomeres shorten over the course of a lifetime. The discovery that chronic psychological stress accelerates this shortening, that the experience of sustained psychological pressure produces measurable physical changes at the molecular level of the chromosome, was significant enough to earn the researchers who made it, Elizabeth Blackburn and her colleagues, the Nobel Prize in Physiology or Medicine in 2009. The mind and the body are not separate systems that occasionally influence each other. They are one system, so thoroughly integrated that the boundary between them is a conceptual convenience rather than a biological reality. What happens in the mind happens in the body. What happens in the body happens in the mind. The emotions you feel change your physiology in measurable ways, not just the dramatic acute emotions of fear and joy but the quieter chronic ones, the sustained emotional tone of a life, the background radiation of how you generally feel about being alive, all of it leaving its mark in the tissue and the chemistry and the cellular machinery of the body you inhabit. The brain structure most associated with the experience of positive emotion, with pleasure and motivation and the drive to seek out the things that sustain and enrich life, is the dopaminergic system, the network of neurons that use the neurotransmitter dopamine as their primary signaling molecule. Dopamine is often described as the pleasure chemical, the brain's reward signal, the substance released when something good happens. This description is not wrong, but it is incomplete in a way that matters. Dopamine is not primarily a pleasure signal. It is primarily an anticipation signal, released not so much in response to receiving a reward as in response to the prediction of a reward, the expectation of something good about to happen. The pleasure of anticipation, the motivating pull of an expected reward, the forward-leaning quality of desire, these are dopamine experiences. The satisfaction of having received what you anticipated is something else, mediated by different neurochemistry, typically briefer and less intense than the anticipation that preceded it. This is why the things we pursue so urgently so often disappoint us slightly once we have them. The dopamine system evolved to motivate the seeking of things necessary for survival: food, water, social connection, safety, and it does this by making the anticipation of those things feel compelling and important, more compelling in many cases than the actual having of them. The wanting is neurochemically more powerful than the getting. And the system is easily hijacked by anything that reliably produces a dopamine spike. Substances like cocaine and amphetamines work primarily by flooding the dopaminergic system with far more dopamine than any natural reward could produce, overwhelming the system with an intensity of anticipatory signal that has no natural equivalent and from which the system struggles to recover. The brain's capacity to be hijacked by its own reward circuitry is a consequence of the same plasticity that makes it capable of learning anything at all. The brain changes in response to experience. This is the fundamental principle of neuroplasticity, the discovery that transformed neuroscience in the latter decades of the 20th century and overturned centuries of assumption that the adult brain was fixed and unchangeable, its circuits set in early childhood and thereafter static. The adult brain is not static. It is continuously remodeling itself in response to what it experiences, strengthening the connections between neurons that are frequently activated together and allowing the connections between neurons that are rarely activated to weaken and eventually disappear. Every skill you learn, every habit you form, every piece of information you encounter changes your brain in a small but real and measurable way, altering the physical structure of the organ in which your entire experience of being alive is generated. The phrase "neurons that fire together wire together," coined by the psychologist Donald Hebb in 1949, though the neuroscience that supports it came later, captures this principle in its simplest form. When two neurons are activated simultaneously or in close succession, the synaptic connection between them is strengthened, making it more likely that the activation of one will trigger the activation of the other in the future. Repeat the pattern often enough, practice a skill, rehearse a memory, reinforce a habit, and the connection becomes so strong that the pathway is activated almost automatically, requiring minimal conscious effort to initiate. This is what a skill being learned feels like from the inside. The awkward, effortful, conscious process of early learning gradually giving way to fluency and automaticity as the underlying neural circuitry is strengthened by repetition. You are not just learning to play the piano or speak a language or ride a bicycle. You are physically rebuilding your brain, adding structure to it, changing its architecture in ways that persist indefinitely. The persistence of learned neural pathways is both the brain's greatest asset and, in certain circumstances, its most challenging feature. The same plasticity that allows skills and knowledge to be acquired and retained also allows fear responses and maladaptive habits and traumatic memories to be encoded and retained with similar durability. The neural circuits of a traumatic memory are strengthened by the same mechanisms that strengthen the circuits of a useful skill, the emotional intensity of the experience ensuring that the connections are formed strongly and maintained persistently, the memory returning with a vividness and an immediacy that can make the past feel, neurologically, like the present. The brain that holds all of this, the skills and the fears and the habits and the memories and the dopamine-driven anticipations and the amygdala's ancient alarm system is generating, right now, a continuous low-frequency electrical oscillation that can be measured from outside the skull. The brain produces different patterns of electrical activity depending on its current state, the rapid, irregular oscillations of alert wakefulness giving way to the slower, more regular waves of relaxed attention, and those giving way in turn to the even slower waves of drowsiness and then sleep. Right now, if you are drifting, if the words are beginning to carry you somewhere slightly softer and slightly less defined than the fully waking state, your brain is producing waves in the alpha range, between 8 and 13 cycles per second, the signature of relaxed, unfocused awareness. The particular quality of attention that arises when the demands of the external world have been set aside and the mind is allowed to simply be, without agenda, without task, without the effortful, directed focus that characterizes waking cognitive work. This is the state the brain finds most naturally restful in wakefulness. This is what is happening in you right now. Let it deepen. Let the waves slow. Let the body continue its extraordinary work in the dark. Consider the eye. Not as a poetic object, not the window to the soul or the mirror of emotion or any of the other roles that literature and philosophy have assigned to it across the centuries of human self-examination. But as a physical structure. A biological instrument of almost absurd sophistication, evolved over hundreds of millions of years from the simplest possible beginning, a patch of light-sensitive cells on the surface of an ancient organism, capable of nothing more than detecting the difference between light and dark, into the extraordinary apparatus that is allowing you, right now, to process these words. The human eye can distinguish approximately 10 million distinct colors. It can detect a single photon of light under ideal conditions, a single quantum of electromagnetic radiation, the smallest possible unit of light, arriving at the retina and being converted into a neural signal that the brain can process. In complete darkness, adapted to the dark, the human eye is sensitive enough that a person standing on a clear night could theoretically detect a candle flame from a distance of approximately 48 kilometers. The eye is not merely a camera. It is a camera of extraordinary sensitivity and range, capable of functioning across a range of light intensities spanning 10 orders of magnitude, from the dimmest starlight to the brightest noon sun, by adjusting the size of the pupil, shifting between two different types of photoreceptors, and continuously recalibrating the sensitivity of the retinal cells to the prevailing light level. The retina that lines the back of the eye contains approximately 120 million rod cells, the photoreceptors responsible for vision in low light conditions, sensitive to brightness but not to color, and 6 to 7 million cone cells, responsible for color vision and fine detail, concentrated in a small central region of the retina called the fovea. The fovea, which is roughly the size of a pinhead, is the only part of the retina capable of resolving fine detail with the clarity that we think of as normal vision. Everything in your peripheral visual field, the large portion of your visual experience that surrounds the small central region of sharp focus, is actually being processed at a much lower resolution than you typically realize, the brain filling in the apparent detail from expectation and memory rather than from actual high-resolution visual data. You see sharply in a much smaller region of your visual field than you think you do. The richly detailed visual world that surrounds you is, in significant part, a construction and interpretation built by the brain from incomplete data, a best guess about what is probably there based on what was there a moment ago and what experience suggests is likely to be there now. The brain constructs your visual experience. This is not a metaphor or a philosophical position, but a literal description of what is happening in your visual cortex right now. The raw data that arrives at the retina is processed through a series of increasingly complex stages as it travels through the visual system, each stage extracting different features, edges, colors, motion, depth, faces, objects, and the final experience of seeing is assembled from all of these process streams simultaneously, a remarkably coherent synthesis of a staggering amount of information happening in the fraction of a second between light hitting the retina and the experience of a visual scene appearing in consciousness. Among the most striking demonstrations of the constructive nature of vision is the blind spot. Every human eye has one, a small region of the retina, approximately 15 degrees from the center of the visual field, where the optic nerve exits the eye on its way to the brain. This region contains no photoreceptors at all. There is a genuine hole in your visual field, in each eye, where no visual information is being collected. And yet you do not experience a hole in your vision. The brain fills it in, smoothly and automatically, using the visual information from the surrounding area to generate a plausible continuation of whatever pattern or surface is present at the edge of the blind spot. The absence is made invisible by the brain's insistence on completeness. You are looking through a hole you cannot see, because the part of your brain responsible for visual experience refuses to show you one. The hearing system is performing a similar feat of construction right now, converting the pressure waves in the air produced by whatever sounds are present in your environment into the experience of sound through a mechanism of extraordinary delicacy and complexity. The ear canal funnels sound waves to the eardrum, a membrane approximately 8 to 9 millimeters in diameter, thinner than a sheet of paper, which vibrates in response to the pressure variations of incoming sound. Those vibrations are transmitted through three tiny bones, the malleus, the incus, and the stapes, the smallest bones in the human body, the stapes measuring just 3 millimeters in length, to the oval window of the cochlea, the fluid-filled spiral structure of the inner ear where the mechanical vibrations of sound are converted into neural signals. Inside the cochlea, the basilar membrane runs along its entire length and is set into motion by the fluid waves created by the vibrating stapes. Different regions of the basilar membrane respond to different frequencies of sound, the base of the cochlea responding to high frequencies, the apex to low ones, and the hair cells that sit on top of the basilar membrane convert its mechanical motion into electrical signals that the auditory nerve carries to the brain. The hair cells of the cochlea are among the most fragile structures in the human body. They do not regenerate when damaged. The hearing loss caused by prolonged exposure to loud noise, the gradual reduction in the ability to hear high frequencies that affects so many people as they age, caused by the cumulative damage to the hair cells at the base of the cochlea, is permanent. The hair cells that are destroyed by a single evening at a very loud concert are destroyed forever, their contribution to the frequency range of your hearing gone without possibility of replacement. The body that replaces its skeleton every 10 years and its skin every few weeks cannot replace a single hair cell in the cochlea. The sense of smell that complements hearing and vision is, in evolutionary terms, the oldest of the human senses. The olfactory system, the neural hardware for detecting and processing chemical signals in the environment, is one of the most ancient parts of the brain, connected more directly to the limbic system and the structures associated with emotion and memory than any other sensory system. This is why smell is the sense most powerfully associated with memory and emotional response, why a particular scent can transport you instantly and involuntarily to a specific moment in your past, producing an emotional response of an intensity and immediacy that no other sensory trigger quite matches. The smell of a specific food, the perfume of a specific person, the particular combination of grass and rain and something else that takes you immediately back to a specific afternoon in childhood. These experiences are not poetic coincidences but the product of the direct anatomical connection between the olfactory system and the hippocampus and amygdala, the brain structures most involved in the formation and retrieval of emotionally significant memories. The human nose can detect approximately 1 trillion distinct odors. That figure, published in a 2014 study in Science magazine that used a systematic combinatorial approach to estimate the discriminatory capacity of the human olfactory system, was considerably larger than the previously assumed figure of around 10,000. And it suggests that smell is not the diminished, rudimentary sense that it is sometimes characterized as in comparison to the visual and auditory systems. The olfactory receptor neurons that line the nasal cavity, there are approximately 6 million of them, expressing around 400 distinct types of olfactory receptor proteins, send their signals directly to the olfactory bulb, a small structure at the base of the brain that processes the incoming chemical information and passes it to the higher brain regions where odor is consciously perceived. The olfactory neurons are among the only neurons in the adult brain that are regularly replaced. New olfactory receptor neurons are generated from stem cells in the nasal epithelium throughout life, one of the very few examples of significant neurogenesis in the adult human nervous system. The sense of touch, the somatosensory system, is distributed across the entire surface of the body through a network of specialized receptors embedded in the skin, each type responding to a specific category of mechanical stimulus. Meissner's corpuscles, concentrated in the fingertips and lips, detect light touch and texture. Pacinian corpuscles, found deeper in the skin and in the joints, detect pressure and vibration. Merkel's discs detect sustained pressure and fine spatial detail. Ruffini endings detect skin stretch. Free nerve endings detect temperature and pain. The density of these receptors varies enormously across the body surface. The fingertips and the lips have the highest density of touch receptors and the correspondingly finest spatial resolution, capable of detecting textures and shapes with extraordinary precision, while the back and the upper arms have a much lower receptor density and a correspondingly cruder spatial resolution. This variation in receptor density is represented in the brain's somatosensory cortex in a distorted map of the body called the homunculus. If you built a figure whose body parts were sized in proportion to their representation in the somatosensory cortex, you would produce a grotesque creature with enormous hands and lips and a tiny torso, its proportions determined entirely by the relative importance the brain assigns to information from each body region. The pain system that operates alongside these touch receptors is one of the most complex and least well understood systems in the body, and one of the most clinically important. Pain is not simply the detection of tissue damage, it is an experience constructed by the brain from multiple inputs, including but not limited to the signals arriving from the site of injury. The same stimulus can produce dramatically different pain experiences depending on context, expectation, attention, emotional state, and the meaning the person assigns to the sensation. Soldiers wounded in battle frequently report feeling little or no pain from their injuries in the immediate aftermath. The context of survival and the intense focus on the immediate situation modulating the pain experience profoundly. The placebo effect, the genuine, measurable reduction in pain produced by an inert treatment believed to be effective, demonstrates that the brain's expectation of pain relief can produce actual neurochemical changes, including the release of endogenous opioids, that reduce the pain experience in ways that are physiologically real rather than merely imagined. The body's own pain relief system, the endogenous opioid system, the network of receptors and their natural ligands that the brain uses to modulate pain, is activated not only by injury and stress but by a range of other experiences including vigorous exercise, laughter, social bonding, and music. The runner's high, the feeling of euphoria and reduced pain sensitivity that sometimes accompanies prolonged aerobic exercise, was long attributed to endorphins, the best known of the endogenous opioids, but more recent research suggests that endocannabinoids, the body's own cannabis-like molecules, which bind to the same receptors as the active compounds in cannabis, may play an equal or greater role. The body is its own pharmacy, producing molecules that manage pain and produce pleasure and regulate mood and modulate inflammation and perform dozens of other functions, using the same molecular keys that plants and fungi have independently evolved to produce, apparently because the locks those keys fit are so fundamentally important that evolution keeps finding them from multiple different starting points. Your body is a pharmacy, a library, a construction site, an electrical grid, a waste management system, a communications network, and a military defense force, all operating simultaneously, all integrated into a single coherent system so seamlessly that you experience it not as the staggering complexity it is but simply as yourself. Simply as being alive. Which is, when you think about it carefully enough, the most extraordinary thing there is. There is a moment in the process of falling asleep, you may have experienced it, perhaps you are approaching it now, when the body gives a sudden involuntary jerk. A twitch, sharp enough to startle you back toward wakefulness, accompanied sometimes by the vivid sensation of falling or stumbling, the physical environment lurching beneath you in a way that feels entirely real for the fraction of a second before the waking mind reasserts itself and reminds you that you are lying down, that there is no edge to fall from, that the ground is exactly where it was when you closed your eyes. This is called a hypnic jerk, or a sleep start, and it happens to the majority of people at some point in their lives. Surveys suggest that between 60 and 70% of people experience them regularly. What causes them is not entirely settled science, but the most widely accepted explanation involves the particular transitional state of the nervous system as it moves from wakefulness to sleep. As the muscles begin to relax in the early stages of sleep onset, the brain, or a specific part of it, the reticular formation in the brainstem, which monitors the state of the body's musculature, may misinterpret the sudden relaxation of muscle tone as a sign that the body is falling. The reflex response is the same one that would be appropriate if you actually were falling, a sudden muscular contraction to catch yourself. By the time the contraction fires, the brain has already realized its mistake, but the twitch has happened, the moment of apparent falling is already over, and you are left lying there in the dark, slightly more awake than you were a moment ago, your heart rate briefly elevated, the dream that was beginning to form already dissolving back into the softer substance from which dreams are made. The body is full of reflexes like this, automatic responses that bypass conscious control, hardwired into the nervous system at a level below deliberate thought, operating on timescales that deliberate thought cannot match. We have already visited the spinal cord reflex that pulls your hand from a hot surface before you know it is hot. But the reflex repertoire of the human body extends far beyond the emergency responses. The gag reflex that protects the airway from objects that enter the throat. the corneal reflex that closes the eyelid when anything approaches the eye too quickly, the patellar reflex that causes the lower leg to kick forward when the tendon just below the kneecap is tapped, the familiar knee-jerk reaction, whose reliability as a diagnostic indicator of spinal cord function makes it one of the most frequently performed tests in all of clinical medicine. The sneezing reflex is one of the more dramatic in the body's repertoire. A sneeze begins with the detection of an irritant in the nasal mucosa, a particle of dust, a fragment of pollen, a pathogen attempting to establish itself in the warm and hospitable environment of the nasal passages by the trigeminal nerve, which sends a signal to the sneeze center in the lateral medulla of the brainstem. The brainstem coordinates a response of remarkable complexity and considerable force. The eyes close. The tongue moves to the roof of the mouth. The muscles of the chest and abdomen contract powerfully, building pressure in the lungs. The soft palate drops. And then the air is released explosively through the nose and mouth at speeds that have been measured at up to 160 km per hour, carrying with it whatever irritant provoked the response, along with a spray of droplets that can travel up to 8 meters from the sneezer's face. The entire process takes approximately 150 milliseconds. It cannot be stopped once initiated. Attempts to suppress a sneeze by pinching the nose or keeping the mouth closed simply redirect the force of the expulsion in ways that can be genuinely dangerous, the pressure that cannot be released externally sometimes causing damage to blood vessels in the eyes or ears or, in rare cases, the brainstem. The body's reflexes are so deeply embedded in the architecture of the nervous system that they persist in circumstances where almost nothing else does. The sucking reflex present in newborns. The grasping reflex that causes an infant's fingers to close around anything placed in the palm. The startle reflex, the Moro reflex, that causes a newborn to fling its arms outward and then bring them together when it feels a sudden loss of support. A reflex that appears to be a vestige of the grasping behavior of our primate ancestors. The infant reflexively reaching to hold on to the mother when it feels itself falling. These reflexes are present from birth, operating before the cortex has developed enough to override them, and they fade as the developing brain matures and the higher control centers come online, a biological timeline written in the disappearance of behaviors that were once involuntary becoming optional and then unavailable as the nervous system grows into its adult architecture. The developing brain that produces these reflexes begins its work extraordinarily early. Neural tube formation, the first step in the development of the central nervous system, begins in the embryo approximately 3 to 4 weeks after fertilization, before most women know they are pregnant, before the embryo has recognizable human features of any kind. By the time a fetus is 25 weeks old, all of the neurons it will ever have are already present, approximately 100 billion of them, formed at a rate of roughly 250,000 new neurons per minute during the peak of prenatal neurogenesis. After birth, no significant new neurons are added to most brain regions. What changes in the years of childhood and adolescence is not the number of neurons but the connections between them, the extraordinary proliferation and then selective pruning of synaptic connections that shapes the brain from the dense, relatively undifferentiated neural network of infancy into the specialized, efficient architecture of the adult brain. The pruning process, the selective elimination of synaptic connections that are not being used, the brain refining its own circuitry through a process that resembles, in some ways, the editing of a text, is one of the most important and least discussed aspects of human development. At its peak, in early childhood, a child's brain contains far more synaptic connections than an adult brain, up to twice as many by some estimates, the neural architecture dense and highly connected, the brain in a state of maximum plasticity and maximum sensitivity to experience. As development proceeds and the brain is exposed to the specific environment and the specific demands of the specific life it is living, The connections that are frequently used are strengthened and the connections that are not are eliminated, the brain becoming more specialized, more efficient, more precisely configured for the particular world it has been growing up in. This pruning process continues into the mid-20s, considerably later than most people realize. The prefrontal cortex, the region of the brain most associated with executive function, with the ability to plan ahead, to inhibit impulses, to evaluate the long-term consequences of short-term decisions, to regulate emotional responses in the light of rational consideration, is the last brain region to complete its development, the final synaptic pruning and myelination of its circuits not complete until approximately the age of 25. The legal and cultural thresholds we use to mark the transition from adolescence to adulthood: 18 in most jurisdictions for voting and military service, 21 in the United States for alcohol consumption, do not correspond particularly well with the actual developmental timeline of the brain structures most responsible for mature judgment. The myelination that completes alongside synaptic pruning is worth its own moment of attention. Myelin is a fatty substance produced by specialized cells called oligodendrocytes that wraps around the axons of neurons in a segmented insulating sheath, dramatically increasing the speed at which electrical signals travel along the axon. An unmyelinated axon conducts signals at roughly half a meter per second. The same axon, once myelinated, conducts signals at up to 120 meters per second, an increase in conduction speed of approximately 240-fold. The progressive myelination of neural circuits during childhood and adolescence is what makes the increasing cognitive sophistication of development possible, the brain not just adding connections but making its existing connections dramatically faster and more reliable. The neural infrastructure being upgraded from the equivalent of a dirt track to a high-speed highway, the same information traveling between the same points but arriving 240 times sooner. The heart that has been beating throughout all of this 100,000 times today, as it beats 100,000 times every day, began beating approximately 22 days after fertilization, before the embryo has a recognizable heart structure at all. The cardiac cells contracting spontaneously and synchronously in a behavior that emerges from the intrinsic electrical properties of the cells themselves rather than from any external instruction. The heartbeat does not begin because the heart is told to beat. It begins because cardiac muscle cells, when they come together in sufficient numbers, simply start beating on their own, the rhythm and emergent property of the cell's collective electrical behavior, arising from the bottom up rather than being imposed from the top down. This intrinsic rhythmicity of the heart, its ability to generate its own electrical impulse without instruction from the brain, is maintained throughout life by a small cluster of specialized cells in the right atrium called the sinoatrial node, the heart's natural pacemaker. The sinoatrial node generates an electrical impulse approximately 60 to 100 times per minute at rest, the impulse spreading from there across the atria and then through a specialized conduction system to the ventricles, the precisely timed sequential activation of the heart's chambers producing the coordinated pumping action that moves blood through the body with each beat. The brain modulates the rate of this rhythm through the autonomic nervous system, slowing it during rest and sleep through the parasympathetic system, accelerating it during stress and exercise through the sympathetic system, but the fundamental rhythm is generated by the heart itself, independently, autonomously, from the first beat 22 days after fertilization to the last beat at the end of life. A rhythm that never stops. A rhythm that is happening right now, in your chest, as these words arrive. The same rhythm that has been there since before you were born, that will be there in the deepest hour of the night when you are most fully asleep, that has accompanied every moment of your life without ever drawing attention to itself, without ever asking to be noticed, without ever pausing for rest. The heart just beats. As it has always beaten. As it will beat tonight, through all the hours of your sleep, through the slow wave phases and the REM phases and the quiet middle of the night when the brain is cleaning itself and the growth hormone is doing its repair work and the memories of the day are being sorted and filed and integrated. The body is taking care of everything. It always has. All it needs from you, right now, is to let it. The stomach is doing something right now that it does every time it is empty for long enough. It is talking. Not metaphorically, the growling sound that an empty stomach produces, the sound that announces itself at the worst possible moments in quiet rooms, is a real acoustic event produced by a real physiological process. The stomach and the small intestine, in the absence of food to process, do not simply wait in passive silence for the next meal. They engage in a housekeeping cycle, a series of strong muscular contractions that sweep through the digestive tract in waves, clearing residual food particles and bacteria and digestive secretions from the walls of the gut in preparation for the next meal. These contractions are called the migrating motor complex, and they occur approximately every 90 to 120 minutes in a fasting person, producing the rumbling and gurgling sounds. The technical term is borborygmi, a word that is itself almost onomatopoeic, that we associate with hunger. The stomach is not simply telling you it is empty. It is actively cleaning itself, maintaining its own hygiene with a thoroughness and a regularity that continues whether you are aware of it or not, whether you are awake or asleep, whether you have eaten recently or not. The digestive system that produces these sounds is one of the longest and most complex organ systems in the human body. From the moment food enters the mouth to the moment its remnants leave the body, it travels through approximately 9 meters of digestive tract: the esophagus, the stomach, the small intestine, the large intestine, each section specialized for a different aspect of the process of breaking food down into its constituent molecules and absorbing those molecules into the bloodstream. The small intestine alone is approximately 6 to 7 meters long, coiled and folded into the abdominal cavity with extraordinary compactness, its inner surface covered in finger-like projections called villi and, on the surface of each villus, even smaller projections called microvilli. The combined effect of this folding and projection increasing the total absorptive surface area of the small intestine to approximately 250 square meters. Another tennis court worth of surface area, folded into another impossibly small space, this time dedicated not to gas exchange but to the absorption of the nutrients that the body needs to maintain itself and generate the energy to do everything else. The process of digestion begins not in the stomach, as most people assume, but in the mouth. Saliva, produced by 3 pairs of salivary glands at a rate of approximately 1 to 1.5 liters per day, contains digestive enzymes, most notably amylase, that begin breaking down the carbohydrates in food before it has even been swallowed. The act of chewing, which seems so mundane as to be beneath notice, is actually a sophisticated mechanical pre-processing step that dramatically increases the surface area of food available to the digestive enzymes, making the chemical breakdown that follows considerably more efficient. A person who chews their food thoroughly extracts more nutrition from the same amount of food than one who swallows it quickly, the body rewarding the small investment of time and attention with a more complete harvest of what the food contains. The stomach that receives the chewed and enzyme-treated food from the esophagus is a muscular bag capable of expanding from a resting volume of approximately 75 milliliters when empty to over 1 liter when full, and in cases of significant overeating, to as much as 4 liters. It churns its contents with powerful muscular contractions while secreting hydrochloric acid strong enough, at its maximum concentration, to dissolve metal and the enzyme pepsin, which begins the breakdown of proteins. The stomach's own cells are protected from this acid by a thick layer of mucus that lines the stomach wall, renewed continuously by specialized mucus-secreting cells. When this protection fails, when the mucus layer is damaged by the bacterium Helicobacter pylori or by prolonged use of anti-inflammatory medications or by other factors, the acid begins to digest the stomach's own lining, producing the painful erosions called peptic ulcers. The stomach is simultaneously producing one of the most corrosive substances in the body and protecting itself from it, moment by moment, with a precision that makes the occasional failure remarkable for its rarity rather than its occurrence. The liver that processes what the small intestine absorbs is the largest internal organ in the body and one of the most metabolically versatile. It performs over 500 distinct biochemical functions, filtering the blood of toxins and metabolic waste products, producing bile for fat digestion, synthesizing the proteins that allow blood to clot, storing glucose as glycogen and releasing it when blood sugar falls, metabolizing drugs and alcohol, producing cholesterol and the lipoproteins that transport it through the bloodstream, generating heat as a byproduct of its intense metabolic activity. No other organ in the body approaches the liver's functional variety. And unlike most organs, which perform their functions continuously at a relatively constant rate, the liver operates dynamically, shifting its priorities and its outputs in response to the body's changing needs, ramping up glucose production during fasting, shifting to fat metabolism during prolonged exercise, prioritizing detoxification after alcohol consumption, adjusting its output of clotting factors in response to injury. The liver is also one of the only organs in the human body capable of significant regeneration. Remove up to 70% of a healthy liver and the remaining tissue will regenerate to approximately its original size within weeks, the remaining hepatocytes dividing and multiplying until the organ has rebuilt itself. This regenerative capacity is the basis for living donor liver transplantation, in which a portion of a healthy donor's liver is surgically removed and transplanted into a recipient whose own liver has failed, both the donor remnant and the transplanted segment then regenerating to functional full size in their respective owners. The liver rebuilding itself from a fragment. Growing back. The body's most spectacular act of self-repair, operating on a timescale of weeks rather than years. The kidneys that work alongside the liver in the body's filtration and regulation system are a pair of fist-sized organs sitting on either side of the spine in the back of the abdominal cavity, each one containing approximately 1 million individual filtration units called nephrons. Together, the two kidneys filter the entire volume of blood in the body approximately once every 30 minutes, about 48 times per day, passing roughly 180 liters of fluid through the nephrons daily, the vast majority of which is reabsorbed back into the bloodstream and only about 1 to 2 liters excreted as urine. The precision with which the kidneys regulate the composition of the blood, maintaining the concentrations of sodium, potassium, calcium, and dozens of other substances within the narrow ranges that allow every other system in the body to function, is one of the most impressive regulatory feats in all of physiology. The kidneys do not merely filter. They also produce hormones. Erythropoietin, produced by the kidneys in response to low oxygen levels in the blood, stimulates the bone marrow to produce more red blood cells, the same hormone that has been infamously abused by endurance athletes seeking to increase their red blood cell count and thereby their oxygen-carrying capacity. Renin, produced by the kidneys in response to low blood pressure, initiates a hormonal cascade that ultimately causes blood pressure to rise, the kidneys acting not only as passive monitors of blood composition but as active participants in the regulation of cardiovascular function. The kidney is not a filter. It is a filter, a hormone factory, a blood pressure regulator, and a red blood cell production controller, all in a fist-sized package sitting quietly in the back of the abdominal cavity, doing its several hundred jobs without drawing attention to itself. The pancreas that sits near the stomach is similarly multifunctional, performing the apparently unrelated jobs of producing digestive enzymes for release into the small intestine and producing the hormones insulin and glucagon for release into the bloodstream with equal competence and equal indispensability. Insulin, released in response to rising blood glucose after a meal, signals cells throughout the body to take up glucose from the blood, lowering blood glucose back toward its resting level. Glucagon, released in response to falling blood glucose, signals the liver to release stored glucose into the blood, raising blood glucose back toward its resting level. The two hormones working in opposition, one rising as the other falls, maintaining blood glucose within the narrow range that the brain, which uses glucose as its primary fuel and has very limited capacity to store it, requires for normal function. Diabetes, in its various forms, is the failure of this system, the inability of the pancreas to produce sufficient insulin, or the failure of the body's cells to respond to the insulin it produces, resulting in blood glucose levels that rise to levels incompatible with normal physiology, with consequences that affect virtually every organ system in the body over time. The endocrine system that the pancreas is part of, the network of glands that produce and release hormones into the bloodstream, is the body's slow communication network, the counterpart to the fast electrical signaling of the nervous system. Where nerve signals travel in milliseconds, hormones travel in minutes to hours, their effects playing out over timescales of hours to days rather than fractions of a second. The thyroid gland in the neck produces hormones that set the metabolic rate of virtually every cell in the body. Too much thyroid hormone and the metabolism runs fast, producing heat and anxiety and weight loss and a racing heart. Too little and it runs slow, producing cold and fatigue and weight gain and depression. The adrenal glands produce not only the adrenaline of the acute stress response but cortisol, the stress hormone whose effects play out over hours and days rather than seconds, modulating immune function and metabolism and memory formation and dozens of other processes in response to the sustained demands of a challenging environment. The body is always listening to its environment, always responding, always adjusting. The hormones that flow through the bloodstream are the medium through which the body conducts its conversation with the world, translating the experiences of a life into the biochemistry of the cells, the outer world becoming the inner world through the extraordinary alchemy of the endocrine system. That conversation is happening right now. In the quiet of this moment, in the slowing of the breath and the cooling of the body and the gathering of the sleep that is almost here, the body is producing the hormones of rest, of repair, of the deliberate, purposeful downshift that happens every night and that is, in its own unhurried way, as active and as extraordinary as anything the body does in the full engagement of the waking day. The stomach is quiet now. The digestive cycle has reached one of its resting intervals. The liver is doing its patient work. The kidneys are filtering. The pancreas is balancing. Everything is in its right place. Everything is working. Everything, right now, is exactly as it should be. Let it be. There is something happening at the very edge of your cells right now that has been happening since before you were born and that will continue until the last moment of your life, something so fundamental and so continuous that it makes the heartbeat and the breath seem almost intermittent by comparison. It is called metabolism. Not in the colloquial sense, not the vague notion of metabolism as the thing that determines whether you gain or lose weight, the mysterious biological variable that some people seem to have more of than others. But in the precise biochemical sense, the sum total of all the chemical reactions happening simultaneously in every cell of your body, the continuous transformation of the molecules you consume into the energy and the structural materials that sustain everything else. Metabolism is not a single process. It is the aggregate of tens of thousands of distinct chemical reactions, each one catalyzed by a specific enzyme, each one producing a specific product that feeds into other reactions, the whole network so interconnected and so finely regulated that disrupting any one of its components tends to send ripples through dozens of others. At the center of this metabolic network is a molecule called ATP, adenosine triphosphate, the universal energy currency of the cell, the form in which the energy extracted from food is stored and transported to wherever in the cell it needs to be used. Every energy-requiring process in the cell, the contraction of a muscle fiber, the pumping of ions across a membrane, the synthesis of a protein, the copying of DNA runs on ATP, the energy released when ATP loses one of its phosphate groups, the molecule converted to ADP and then regenerated back to ATP in a cycle of extraordinary efficiency and extraordinary speed. A resting human body produces and consumes its own body weight in ATP approximately every 24 hours. Not the weight of the ATP itself, the weight of your entire body. Every cell in your body is cycling through its own weight in ATP every single day, the molecule being made and used and remade thousands of times per cell per day, the currency of cellular energy changing hands at a rate that makes any financial market look sluggish. The ATP is produced primarily in the mitochondria, the small organelles present in virtually every cell in the body, often described in the shorthand of popular science as the powerhouses of the cell. The description is accurate but insufficient. The mitochondria are power generators, yes, but they are power generators of a very specific and very remarkable kind. They are, in the most literal biological sense, the descendants of free-living bacteria that were incorporated into the cells of an ancestral organism approximately 1.5 to 2 billion years ago, in one of the most consequential events in the history of life on Earth. A bacterial cell, engulfed by a larger cell rather than digested, retained inside the host cell and eventually becoming so thoroughly integrated into its biology that the two organisms are now inseparable, the mitochondria still carrying their own DNA, still dividing independently of the cell that contains them, but no longer capable of surviving outside it, the ancient bacterial independence long since traded for the security of integration. Your mitochondria are not entirely yours. They are the descendants of bacteria. Ancient, fully integrated, indispensable bacteria, but bacteria nonetheless, their separate origin detectable in their separate DNA, in the fact that mitochondrial DNA is inherited exclusively from the mother rather than from both parents, in the structural details of the mitochondrial membrane that still echo the membrane of the free-living bacterium it once was. The power generators running every cell in your body are ancient bacterial immigrants, so thoroughly domesticated by 2 billion years of coevolution that their foreignness is invisible, but foreign in origin nonetheless. You are, at a fundamental cellular level, a collaboration. A partnership between different lineages of life, merged so completely that the seam between them has become invisible. The DNA that the mitochondria carry, the mitochondrial genome, a small circular molecule containing just 37 genes, is almost entirely separate from the nuclear DNA that carries the vast majority of your genetic information. The nuclear genome, the DNA distributed across the 23 pairs of chromosomes in the nucleus of each of your cells, contains approximately 3 billion base pairs encoding somewhere between 20,000 and 25,000 protein-coding genes. 3 billion base pairs of genetic information, organized into chromosomes and packaged into a nucleus approximately 6 micrometers in diameter, a space smaller than the width of a human hair, through a feat of compaction so extraordinary that if you could uncoil all of the DNA in a single cell and lay it end to end, it would stretch for approximately 2 meters. 2 meters of DNA, folded with extraordinary precision into a space invisible to the naked eye, encoding the instructions for building and running a human being. If you uncoiled all of the DNA in all of the cells in your body and laid it end to end, The resulting strand would stretch for approximately 2 times 10 to the power of 13 meters. 2 followed by 13 zeros. Long enough to stretch from the Earth to the Sun and back approximately 67 times. The genetic information contained in a single human body, if it could be physically extended, would reach the Sun 67 times over. The genome that contains all of this information is not a static document. It is being read right now, in every cell in your body, different genes being transcribed and translated in different cells according to the specific requirements of each cell type, the genes for producing hemoglobin being read in developing red blood cells, the genes for producing insulin being read in the beta cells of the pancreas, the genes for producing the proteins of the lens of the eye being read in the lens cells. The same genome, containing the same 3 billion base pairs, is being interpreted differently in each of the 200 or so distinct cell types in the human body. The differences between a liver cell and a neuron and a muscle cell and a red blood cell arising not from differences in the genome itself but from differences in which parts of it are being read in each cell type and in what quantities. This selective reading of the genome is regulated by a layer of biological control that sits above the genome itself, the epigenome. The system of chemical modifications to DNA and the proteins around which DNA is wrapped that determine which genes are accessible to the cellular machinery for reading and which are not. The epigenome is the reason that genetically identical twins, who share the same DNA sequence, can develop measurable differences in gene expression over time, their different experiences, different environments, different diets and habits and stresses leaving marks on the epigenome that alter how their shared genome is read, the same text being interpreted differently by different readers in different circumstances. The genome that is being read in your cells right now is not the genome you were born with, in one important sense. It has been accumulating changes since the moment of fertilization, small mutations arising as DNA is copied and not perfectly repaired, the tiny inevitable imperfections of a copying process operating at enormous scale and considerable speed. The cells of an elderly person contain on average thousands more mutations than the cells of a newborn, the accumulated errors of a lifetime of cell division, the molecular record of a life written in the language of nucleotide changes. Most of these mutations are harmless, occurring in non-critical regions of the genome or in genes that are not being actively expressed in that cell type. A small number are harmful. And a very small number, the mutations that arise in genes that regulate cell division and cell death, are what we call cancer. Cancer is not a foreign invasion. It is not something that enters the body from outside. It is the body's own cells, altered by mutation to behave in ways that serve the individual cell at the expense of the organism, dividing without the usual constraints, evading the immune surveillance that normally detects and destroys abnormal cells, consuming resources and space in ways that disrupt the function of normal tissue. It is the breakdown of the cooperation that multicellular life depends on, individual cells defecting from the collective enterprise of the organism, reverting to a more primitive mode of existence in which the interests of the individual cell take precedence over the interests of the body that contains it. The immune system that normally prevents this is performing cancer surveillance right now, scanning the surfaces of cells for the molecular signatures of malignant transformation, destroying cells that display those signatures before they can proliferate into tumors. Every day, in a healthy body, the immune system detects and destroys dozens or hundreds of cells that have undergone mutations sufficient to initiate malignant transformation. Cancer arises when a cell acquires mutations that allow it to evade this surveillance, to disguise its abnormality, to suppress the immune response in its immediate environment, to become, in effect, invisible to the system that would otherwise destroy it. The cells that make up your body have been cooperating in this enterprise for the entire length of your life. 37 trillion of them, each one in possession of the full instructions for building a complete human being, each one expressing only the small fraction of those instructions appropriate to its specific role, each one subordinating its individual interests to the collective project of the organism. The cooperation is so complete, so seamless, so embedded in the fundamental biology of every cell that we do not notice it, we notice only ourselves, the unified experience of being a single person, with no awareness of the vast and continuous negotiation between trillions of cells that produces that experience. The body is a democracy of extraordinary complexity, a negotiation of extraordinary delicacy, a collaboration so successful and so ancient that it feels, from the inside, like nothing at all. Like simply being here, in the dark, breathing, your heart beating, your mitochondria humming, your genome being read in 37 trillion cells simultaneously, the whole extraordinary enterprise of you continuing without interruption, without complaint, without any need for your awareness or your gratitude or your conscious participation. The body is taking care of everything. It has always taken care of everything. Rest inside it. Consider for a moment what it means to remember something. Not the experience of remembering, not the feeling of a memory arriving, the particular quality of recognition that accompanies the return of something known, the way a face or a smell or a snatch of music can bring a moment back with an immediacy that makes the distance between then and now seem briefly to collapse. But the mechanism, the actual physical process by which an experience that happened in the past, that is, by definition, no longer happening, leaves a trace in the present that can be retrieved and re-experienced in some form by the person who originally had it. Memory is not a recording. This is perhaps the most important thing to understand about it, and the thing most at odds with the intuitive model that most people carry around in their heads. We tend to think of memory as something like a video file, a stored representation of an original experience that can be played back more or less faithfully, the fidelity perhaps degrading over time but the basic character of the stored information remaining essentially intact. This model is wrong in ways that are both intellectually fascinating and practically significant. Memory is not a playback. It is a reconstruction. Every time you remember something, you are not retrieving a stored copy of the original experience. You are rebuilding it, in the moment of remembering, from the fragments of information that were encoded at the time, filled in with expectations and knowledge and the current emotional state of the rememberer, shaped by everything that has happened since the original experience occurred. The reconstruction is so seamless, so automatic, so convincing that it feels like retrieval. It feels like playing back a recording. But the research of memory scientists, Elizabeth Loftus in particular, whose decades of work on the malleability of human memory has produced findings that have transformed the legal system's understanding of eyewitness testimony, has demonstrated unequivocally that human memory is far more malleable, far more susceptible to revision and distortion and even wholesale fabrication than most people believe or are comfortable believing. False memories, detailed, confident, emotionally resonant memories of events that never happened can be implanted in a significant proportion of research subjects using nothing more than suggestion and leading questions. People have been convicted of crimes on the basis of eyewitness testimony later shown to be entirely false, the witness not lying but genuinely misremembering, the reconstruction that felt to them like retrieval being a construction rather than a copy, built from the fragments of what they actually saw and the expectations and suggestions and subsequent information that shaped the reconstruction into something that bore only a partial resemblance to the original event. The hippocampus, the seahorse-shaped structure buried in the medial temporal lobe, one on each side of the brain, is the structure most central to the formation of new declarative memories, the memories of facts and events that can be consciously recalled and verbally described. It is not where memories are ultimately stored. Long-term memories are distributed across the cortex, stored in the same sensory and association areas that were active when the original experience occurred. The memory of a visual scene stored partly in visual cortex, the memory of a sound stored partly in auditory cortex, the whole memory distributed across the brain like a constellation of related activations that can be reconstructed when any one of them is triggered. But the hippocampus is where new memories are initially formed, where the various sensory and emotional components of an experience are bound together into a coherent episode, where the index is kept that allows the distributed cortical memory to be retrieved and reconstructed. Damage the hippocampus and new memories cannot be formed. The experience of the present moment flows through awareness without leaving a trace, each moment complete in itself and then gone, the world perpetually new and perpetually unfamiliar. The most famous case of hippocampal damage in the history of neuroscience is that of a man known for decades by his initials, H.M. Henry Molaison, whose full name was revealed only after his death in 2008. In 1953, at the age of 27, Molaison underwent surgery to treat severe epilepsy, a bilateral removal of the hippocampus and surrounding medial temporal lobe structures that successfully reduced his seizures but produced a side effect that no one had anticipated. He lost the ability to form new long-term memories. For the remaining 55 years of his life, Molaison lived in a permanent present, able to recall events from before his surgery with reasonable clarity, able to maintain a conversation normally for as long as the conversation continued, but unable to retain any new information for more than a few minutes. Every day was the first day. Every person he met was a stranger, even if he had met them hundreds of times before. Every meal was new. Every piece of news was news he was hearing for the first time. The scientists who studied him had to reintroduce themselves at the start of every session, regardless of how many years they had been working with him, and he greeted each introduction with the same polite interest, the same genuine unfamiliarity, the face of a person meeting a stranger rather than a friend. What Molaison's case revealed, and it revealed it with a clarity and a completeness that would have been impossible to obtain from any other source, was the distinction between different types of memory, and the different brain structures that support them. Because despite being unable to form new explicit, declarative memories, Molaison could still learn. He could acquire new motor skills, improving at mirror drawing tasks, for instance, a task that requires learning to guide a pencil by watching its reflection rather than the pencil itself, over repeated sessions, even though he had no memory of having practiced before. Each session felt to him like the first time. And yet his performance improved, session by session, the skill accumulating in a part of the brain, the basal ganglia and the cerebellum, structures associated with procedural rather than declarative memory that his surgery had left intact. He was learning without knowing he was learning. Remembering without being able to remember. The body acquiring knowledge that the conscious mind had no access to. This distinction between explicit and implicit memory, between the memories we can consciously recall and the memories embedded in our skills and habits and the automatic behaviors that guide so much of our daily life, is one of the most important in all of cognitive neuroscience, and it has profound implications for how we understand what we are and how we know what we know. A very large proportion of what you know, you cannot tell anyone. It is embedded in the procedures your body follows without conscious direction, the way you balance on a bicycle, the way your fingers find the right keys when you type, the way you navigate a familiar route without consciously planning each turn. This knowledge was learned, is retained, and guides behavior, but it does not exist as a retrievable memory in the way that your knowledge of your own name or the events of your childhood does. It exists as a pattern, a tendency, a disposition of the nervous system to behave in a particular way when particular conditions arise. The cerebellum, the small, densely folded structure at the back of the brain, sometimes called the little brain, is the structure most involved in this procedural and motor learning, and it is a remarkable structure in its own right. Despite accounting for only about 10% of the brain's total volume, the cerebellum contains approximately 50% of all the neurons in the brain, more neurons packed into that small folded structure than in the entire rest of the brain combined. This extraordinary neuronal density reflects the computational demands of the cerebellum's primary function, the fine-tuning of movement, the continuous comparison of intended movement with actual movement, the detection and correction of errors in real time, the automation of learned motor sequences. Every skilled movement you make, every stroke of a pen, every step on an uneven surface, every catch of a thrown object is being monitored and refined by the cerebellum, the errors detected and corrected so quickly and so automatically that the correction is complete before you are aware that an error was made. The language system that allows you to understand these words is distributed across the left hemisphere of the brain in most right-hand people, and in a significant proportion of left-handed people as well, the lateralization of language being one of the most robust findings in all of neuropsychology. Broca's area, in the left frontal lobe, is involved in language production, the planning and execution of speech, the selection and sequencing of words, the grammatical structuring of what is to be said. Wernicke's area, in the left temporal lobe, is involved in language comprehension, the extraction of meaning from heard or read language, the decoding of the signal into the understanding of what it means. The two areas are connected by a bundle of nerve fibers called the arcuate fasciculus, and damage to different points in this system produces different and highly specific language impairments, the inability to speak fluently while comprehension remains intact, or the ability to speak fluently while producing language that has no coherent meaning, or the loss of specific categories of words while others remain accessible. The brain's organization of language into specific anatomical regions, with specific functions that can be dissociated from each other by specific patterns of damage, is one of the clearest demonstrations of the principle that mental functions are implemented in physical structures, that the mind is not a ghost in the machine but the machine's own activity, distributed and localized and dependent on the integrity of specific biological structures in ways that become brutally apparent when those structures are damaged. And yet the brain is also capable of a degree of reorganization in response to damage that continues to surprise the researchers who study it. A child who loses the left hemisphere entirely in early life, through stroke or surgery to treat intractable epilepsy, can develop language in the right hemisphere, the functions normally lateralized to the left migrating to the surviving tissue with a completeness that allows near-normal language development. The adult brain is less plastic in this respect, the reorganization possible after damage in adulthood is more limited, the critical periods during which the developing brain is maximally flexible for certain types of learning having long since closed. But even the adult brain reorganizes in response to experience and to loss. Musicians who play stringed instruments show measurable expansion of the cortical representation of the fingers of the left hand, the hand that does the complex fingerwork on the strings. People who become blind in adulthood show their visual cortex recruited for processing tactile information, the same neurons that once processed visual input repurposed for reading Braille, the brain's real estate never lying fallow for long. The brain you are resting in right now is reorganizing itself. Not dramatically, not in the way a recovering stroke patient's brain reorganizes over months of rehabilitation, but quietly, incrementally, in the way that the brain always reorganizes during sleep, the synaptic connections active during the day being evaluated and adjusted during the night, some strengthened and some weakened, the architecture of the brain being subtly refined by the experience of the day in ways that will be imperceptible tomorrow but cumulative over a lifetime. You are not the same person who began listening to this tonight. You are very slightly different, very slightly reorganized, very slightly refined, a few new connections strengthened by the passage of information through them, a few old ones weakened by the competition of new ones. The brain is always making you new. Even now. Even as you sleep. There is a question that has followed human beings for as long as human beings have been capable of asking questions. What am I? Not in the philosophical sense, not the question of meaning or purpose or the place of a human life in the larger scheme of things, though those questions are real and important and have occupied the best minds in every civilization that has ever existed. But in the more immediate, more physical sense. What is this thing I am living inside? What is actually happening in here, in the warm dark of the body, in the space between the first breath and the last? What is the nature of the process that I call being alive? We have been exploring that question tonight. Moving through the body the way you might move through a city you have lived in for years but never fully examined, finding in the familiar streets and buildings a complexity and a richness and a strangeness that was always there, that has always been there, that was simply waiting for the attention that would make it visible. And there is still more. There is always more. The body is not a subject that yields its depths quickly or completely. It is a subject that rewards exactly the kind of patient, unhurried attention that the middle of the night makes possible, the kind of attention you are bringing to it right now, in the soft borderland between waking and sleep, where the mind is loose enough from its ordinary concerns to receive something new. So here, in the last of the quiet, are the final threads. The immune system that we have already visited in its role as cancer surveillance and pathogen defense is also, in its most intimate and most personal dimension, the system responsible for one of the most fundamental biological facts of human existence, the fact that you are you and not something else. Every cell in your body displays on its surface a set of protein markers called MHC molecules, major histocompatible complex molecules, that function as a kind of molecular identity card, a biological signature unique to you, recognized by your immune system as self and therefore left alone. Cells that do not display the right signature, bacteria, viruses, cancer cells that have altered their surface proteins, transplanted cells from another person's body, are recognized as non-self and targeted for destruction. The immune system is not only a defense against the outside world. It is the mechanism by which the body maintains the boundary between self and not self, the biological basis of individual identity at the cellular level. This molecular identity has a chemical dimension that extends beyond the cells to the volatile compounds released by the body into the air, the subtle chemical signature that each person emits through their skin and their breath and their secretions, a signature so specific that trained dogs can identify individuals by scent alone, can track a specific person through a crowd, can detect the chemical changes associated with specific diseases and emotional states with a sensitivity that no human built instrument has yet been able to match. You have a smell that is uniquely yours, produced by the particular combination of your genetics and your microbiome and your diet and your health and the specific circumstances of your life, a chemical autobiography written in volatile molecules and readable, by the right nose, in the air around you. The genetics that contribute to this individual scent signature are the same genetics that appear to influence mate selection in ways that operate below the level of conscious awareness. Studies in which participants were asked to rate the attractiveness of odors from t-shirts worn by strangers have consistently found that people tend to prefer the scent of individuals whose MHC profile is different from their own. The immune system's identity markers apparently providing a chemical signal that the olfactory system can detect and that the brain interprets as attractiveness, steering individuals toward partners whose immune genetics complement rather than duplicate their own. The offspring of such pairings potentially benefiting from a broader and more effective immune repertoire than the children of two parents with similar MHC profiles. The body choosing its partners, at a level below conscious deliberation, for immunological reasons. The hormones that mediate the experience of social bonding, of love and attachment, and the particular comfort of close physical proximity to someone you trust are among the most studied and least fully understood molecules in all of neuroscience. Oxytocin, produced in the hypothalamus and released from the pituitary gland, is often called the love hormone or the bonding hormone, a simplification that does justice to neither the complexity of the molecule nor the complexity of the experiences it is associated with. Oxytocin is released during physical touch, during sexual intimacy, during breastfeeding, during positive social interactions of many kinds, its release associated with feelings of trust and closeness and the reduction of social anxiety. But it is also released in contexts of in-group bonding in ways that can simultaneously increase hostility toward out-group members, its social effects more nuanced and more context-dependent than the simple love hormone label suggests. The molecule that makes you feel close to those you love may also, under certain circumstances, make you feel more suspicious of those you do not know. The social brain that produces and responds to these hormones is the most recently evolved part of the human brain. The large prefrontal cortex and the expanded association areas that distinguish the human brain from those of other primates being the product of an evolutionary expansion that happened relatively recently in geological terms, over the past 2 to 3 million years. The driving force behind this expansion, the selective pressure that made bigger, more socially sophisticated brains advantageous enough to be worth the enormous metabolic cost of running them, is believed by many evolutionary anthropologists to have been the complexity of social life itself. The demands of navigating the intricate social hierarchies and coalitions and relationships of human groups, of modeling the mental states of other individuals, of cooperating and competing and communicating in the ways that human social life requires, may have selected for the cognitive abilities that make us what we are more powerfully than any other environmental pressure. We became smart, in other words, primarily by becoming social. The brain that can understand these words, that can hold the thread of a long narrative, that can imagine the mental states of other people and predict their behavior and feel something about their experiences, that brain is a social organ, shaped by and for the demands of human relationship, its most distinctive capabilities emerging from and serving the fundamental human need to live in community with others. The skin that holds all of this together, the boundary between the internal world of the body and the external world it moves through, is not merely a physical barrier. It is a social organ too. Touch is the first sense to develop in the human embryo and the last to fail in old age, and its role in human social life and human psychological well-being is considerably more fundamental than its status as one sense among many suggests. Premature infants who are held and stroked gain weight faster and leave the hospital sooner than those who receive equivalent medical care without physical contact. Elderly people who receive regular physical touch show better immune function and better cognitive performance than those who are touch-deprived. The simple physical fact of being held, of another body's warmth and pressure against the skin, produces neurochemical changes that reduce stress hormones, lower blood pressure, improve immune function, and promote the kind of parasympathetic nervous system dominance that is associated with rest and recovery rather than threat and response. The body is a social body. It was built for contact, built for the proximity of other bodies, for the particular neurochemical environment that social connection produces, for the regulatory influence that close relationships exert on the autonomic nervous system, the heart rate and the breathing and the stress hormones of two people in close, trusting proximity subtly synchronizing with each other, each person's physiology being gently regulated by the others, the bodies in conversation in a register below language, below conscious awareness. In the ancient wordless dialect of nervous systems that have learned to trust each other. Loneliness, the state of being without this connection, of being physiologically isolated from the regulating influence of close social bonds, has measurable effects on physical health that are comparable in magnitude to the effects of smoking 15 cigarettes per day. Not metaphorically comparable. Statistically comparable, in terms of the increase in mortality risk associated with chronic social isolation versus the increase in mortality risk associated with heavy smoking. The body needs other bodies. It is not designed for isolation. The pain of loneliness is not merely psychological, it is the body registering, through the same neural machinery that registers physical pain, that something essential is missing. That a need as fundamental as food or water or sleep is going unmet. You are not a self-contained system. You never were. You are a node in a network, a point in a web of relationships that extends in every direction, to the people who are physically present in your life, to the people whose influence persists in you long after they are gone, to the ancestors whose genetics you carry and whose epigenetic marks may still be shaping your biology, to the microbiome that has been with you since birth and that is, in a very real sense, part of you. You are more porous, more continuous with your environment and the people in it, more fundamentally relational than the Western tradition of thinking about the self as a bounded, independent individual quite captures. The body knows this. Has always known it. It is written into the neurobiology of social bonding and the physiology of touch and the immunology of self and non-self and the epidemiology of loneliness. The body reaches toward other bodies, regulates itself in their presence, suffers in their absence, knows, at a level far below conscious thought, that to be alive is to be connected, that the self is not a fortress but a conversation, not a fixed point but a process, not a noun but a verb. You are not a thing. You are a happening. A continuous, dynamic, extraordinarily complex process of becoming, sustained by the food you eat and the air you breathe and the water you drink and the sleep you take and the people you love and the microorganisms that live in your gut and the mitochondria that were once bacteria and the immune system that knows you by your molecular signature and the brain that is reorganizing itself, right now, in this moment, in response to everything that has happened today and everything that is happening now. You are the most complex thing in the known universe. And you are resting. And the resting is part of what you are. The breath is slowing. The heart is steady. The brain is beginning its night work. The body is doing what the body does, sustaining itself, repairing itself, preparing itself, making itself ready for the next day's particular demands, the next day's particular joys, the next day's particular version of the endless miracle of being alive. Let it. You do not need to do anything. You do not need to be anything other than what you already are. The body is enough. It has always been enough. It is, by any measure, more than enough. Sleep well.